Good morning, everyone. I'm going to introduce to you a tool that we have been developing and working on for some time now, and we are updating it and improving it in the framework of the integration approach that we have been talking about during these days. So this tool is for single-cell modeling, and I'm going to talk about the workflow that we are following in order to achieve this task. So the general principle is very simple. We have a user that is working in the collaboratory, and this user wants to optimize a neuron model of a single cell starting from the data. So against this data, he wants to optimize the parameters of the model, and then finally he wants to run the simulation of the model itself. If you go down into a bit more detailed view, we see that this can be divided into basically 3 steps. So the first step is the data. So regards the data and more specifically feature extraction. So we start from electrophysiological traces, and we want to extract some properties of these traces that we call features. So basically we have the data, we select the features that we want to extract, we will see which these features are in a few minutes, and we run the feature extraction, and finally we are provided with some results of this step that we can feed to the second step, which means the optimization step. So the second step is the optimization. We select a model with a given morphology, with a behavior that we somehow know, and we use the features that we just extracted to optimize the parameters of this model. So this operation takes time and resources, so we submit this job to an HPC system and we run the optimization. Once the optimization step is finalized, we fetch the results back, and finally we'll run the simulation of this model. So in order to do this, we have something that is going on behind the scenes. So for the features and the data set that we are accessing and the models, we rely on the model catalog, and we are integrating the data fetching with the knowledge graph itself that some speakers talked about yesterday and 2 days ago. So this is the first part of the integration in this workflow. And the same holds for the HPC platform. So this tool that we have built relies on HPC systems. We submit our optimization to HPC, and finally we fetch the results. And we also integrate this workflow with some tool that is already in the brain simulation platform of the collaboratory, which is the Neuron as a Service simulator, and we use for the simulation part, and also we use another web application that we integrated in the brain simulation platform to extract the features. So this is what basically we call the integration part. So we have our web application, which is an interface to provide all these steps and operations to the users, and we integrate with tools, platforms, systems that are doing their job elsewhere, but we try to perform the operation in such a way that this integration is seamless. Okay, so let me show you the tool. The tool is called the Hogdkin-huxley Neuron Builder, and it is accessible from the brain simulation platform. So this is still Collaboratory 1. We are in the process of migrating all our web application and the brain simulation platform itself in Collaboratory 2. And you will have basically the same structure. So we collected the tools of the brain simulation platform in what we call online use cases, which are groups of realistic scenarios that you can encounter while you're doing your research. And we grouped these by families. So we go from the molecular level to the brain area, for example, modeling. And if you go to the single cell building, you have this first online use case, which is called Hogdkin-huxley Neuron Builder, this one. So if you click, this is a web application, so front-end HTML, CSS, and JavaScript, and the back-end is done in Django, which is basically Python. And we want the user to take these tools and copy them or integrate them into their collabs. So we have this functionality here. You can create your own collab. For example, CodeJam 10. And once you click here, this tool is copied in this new collab. So you have the collab here. Okay, so you are provided with this very first interface. You can initiate a new workflow, or you can, while in the process of completing your workflow, you can store the results of the workflow that you have reached till a certain point to the storage of the collaboratory, so you can fetch the ongoing finalized workflow anytime. I will go with this create a new workflow, and here basically we have the 3 steps that I've been talking about. So the workflow has a unique ID, so you cannot submit an optimization in a, let's say, not unique way. So one workflow, one optimization submission. This is to prevent people to have different optimization or workflow that corresponds to different ideas. And the first 3 steps are, the first 2 steps are basically collected here in the cell optimization. So as I was saying before, the first step relates to data, more specifically feature extraction. This web application is called feature extraction graphical user interface. It is reading the data from the data set. So it's taking some time, and here you have the data with some metadata here, the contributors, the species, and so on and so forth. Let's say that I want to select one electrical type, which is a continuous accommodating cell, and I have one unique choice of this. So I have only interneuron and CA1 region of the hippocampus that correspond to this electrical type. So I select this one, then I click on apply, and I am provided with all the data to perform this first step. So these are the traces, and now I want to select the stimulus that correspond to what I'm interested in. For example, the strongest stimuli I would say that produce some spike, like these ones. For example, for this trace and for this trace as well. Okay, I have 3 of them, and then I click next, I confirm, and I'm provided with this interface to select the features of these traces that I'm interested in. So I have 3 types of features, spike events, and then spike shape, and then voltage features. For this step, we rely on a Python package which is called FL, and it's been developed at EPFL, mostly by Werner van Geit. It is available on GitHub, so everything up here you have a link that you can follow to get this package. Okay, so I select this some of the features. This really depends on your experimental protocol, your model, so the features that you want to select for this step. Let's say I just select this one for demonstration purpose, and then I click next. So the system is extracting the features, generating the results, and here are the results. I can download them in case I'm just interested in the features, or like in this case, I can save these features to my workflow. So I click on Save to Workflow here, and here you can see that this bar is green now. So this step has been completed, and I have to do to complete the other ones in order to complete the workflow. On the top bar here, you have the workflow ID, as I was saying before, and then you have also this button. So home, you go home again, you start your workflow, or you select save the workflow from scratch. You can start a new workflow here, and you can save here to the storage. So if I click on storage here, it is saving on the collab storage. So if I go back tomorrow to my collab, I can fetch basically this same workflow with these features already saved there. Okay, this is for the user not to wait until the end of the workflow every time, so that he can interrupt whenever he wants and then come back. And you may also want to start another workflow, but with the same data, because, for example, you want to submit the optimization to 2 different HPC systems. So in this case, you click clone here, and if you can see the workflow ID here, when I click clone, it just changed. Okay, this is unique and allows you to have as many workflows as you want that have unique data in them. Okay, so this is the step related to data. The second step is basically the choice of the model that I want to optimize. It is taking some seconds because the system is loading the model from the model catalog. So we have already stored many models in the model catalog, and these are the hippocampus model. You have an idea of how the morphology of this model is and how the model behaves, actually. So basically, you scroll down, you just select the model that you prefer, like the first one, and this model is stored in my workflow. So I can go on. Of course, you may have your own feature files that you want to upload, or you may have your own model that you want to optimize. So you are provided with this functionality here: upload files for the features, upload files for the optimization. Once you have chosen the model there, you can download it. So it is also available for download if you want to know and see what you're working with, or you can delete it, right, if you want to choose another one. Okay, so now I have basically everything that I need to submit my optimization job, and now I have to select the HPC system that I want the job to run in. I click here, and basically I have 3 choices. So 2 choices are for people who already have credentials on HPC systems. These are CSCS Daint and the Neuroscience Gateway platform. So I have username and password on Neuroscience Gateway. I belong to a project on CSCS Daint. So when I select one of them, I can just submit my job with my own user. But we've been thinking about users, like for example students, that do not have any credentials on HPC systems but may want to submit optimization jobs. So in collaboration with CSCS, and also with Neuroscience Gateway, but Neuroscience Gateway will be here in this list in a few weeks as well, but the principle is exactly the same. So in collaboration with CSCS, we added this functionality which is called service account. And I will show you some slide on this to explain briefly what the service account is about. So the idea is that we have our web application. In my case, I mean, in this web application, you may find this also in the Jupyter notebooks in the collaboratory, where a user with HPC credentials can basically perform the choice that he wants, but a user without any credential at least has one choice, this one, the service account. So the way it works is we created a REST API service that takes the request that the user, on his behalf, the developer, may do for submitting a job. So he's requesting something to us, to our system, and is wrapping the request in a certain way that the token that you get when you enter the collaboratory is provided with the request. So we basically read this request, we store some information about the user ID, the time of job submission, the type of submission that is submitting on our system for tracking what is going on, and then we submit to HPC systems with this service account that is linked to one of our accounts, so to one physical person that has already credentials on that system. So we submit as it was me, but we store the information of the user that requested me to do this. Okay, so it's a basic principle of service account. And the code of this is available on GitHub in the Human Brain Project repository. And in the Brain Simulation Platform guidebook, you have the guidebook, so all the API and how you can perform your request through the service account. If you go just a little bit into the details of the workflow here, we are in the collab, we request with the token, we verify the token thanks to the OIDC system of the collaborator. So now at this point we know who the user is, then we store the data as I was saying before, submit the job, get the results, update the database, for example, with the time of the end of the job, and the status of the job, if it has been canceled, for example, if it's been successful, and so on. And finally, we return the response to the user. We have a small database that has been implemented for storing all this information in a virtual machine that is running on Cineca. We have designed this, but the coding part is basically Roberto, who is here. So if you have any question, please come to us and ask for any doubts that you may have on how we implemented this service account functionality. And this is basically an idea of how we built it. So this is a job descriptor for CSCS through the Unicore interface. This is a job descriptor for NSG. And we just created this very similar job descriptor, but that contains in the headers of the request also the token, so that we can take this one, we unwrap this, we create the Unicore request, and then finally we launch the request on the HPC system. Okay, so we can go back to our workflow. Now I have the service account choice. I choose the default parameters, I click on apply, and the job is submitted. Okay, so, oh, sorry, not yet submitted. So the job is ready to be submitted, so everything is green here. The button is enabled, not job submitted. I click on launch optimization. Now it sends everything to CSCS. And this is done. Okay, so last step. Let's say that we have finalized our optimization. Now we want to fetch the results back to run our simulation. I click on this button. Again, here I can upload a model that I may want to simulate without fetching it from the HPC system, or like in this case, I want to fetch the model, the optimized model that I just submitted, for example. I apply, and I'm provided with the list of all the model that of all the jobs that I've submitted to the service account. So I had a few here, and here basically I have the list of all my jobs. Once I fetch the job, it takes some time to not really to fetch it, but to run some analysis to show you PDF with an example of electrophysiological activity of the model. So it takes like 5 or 6 minutes in general, maybe 1 or 2 more, but not more than 10 minutes in general. So I will not go on fetch now, but I will show you basically the same behavior by uploading a model. So I have a model here that is, for example, this one, which has the same format, a .zip file that I basically reconstruct once I fetch the results of the optimization step. I click on Run Simulation, and what is happening here is basically a procedure thanks to which I'm passing this optimized model to a tool that is also available on the brain simulation platform and is called Neuron as a Service. This has been developed originally by Robert McDougall at Yale, and then the Jean-Denis Courcol's group, Gary Kivaska more specifically, took this code and built an interface on the same principles of the original Neuron as a Service. Here I can visualize the morphology of the model. I can also have a dendrogram for this. And if I zoom in, I can select basically the compartments from which I can record. Here I have all the details of basically my entire model. And here I have the simulation parameter. So let me just reduce this a little bit because I cannot, yeah. So you can set the temperature, the holding current if there's any, T-stop, delay, and whatever, for example, the stimulus amplitude here, and finally you can run the simulation. We are recording from the SOMA, so we will visualize here the traces corresponding to the activity recorded at the SOMA level. Start here in simulation, and this is going on. Okay, so I have a bunch of spikes here. It was, I think it was like 600 milliseconds of stimulation, and 1 second the entire simulation. I can zoom in for more details on the action potential, for example, and also if you want to download the activity, you can click on this cloud here and say all the activity with timestamp and spike neuron, number indices of the neurons in a TXT format. Okay, I think I told you basically everything that I wanted to with respect to these. Of course, this is not a one-person work, but we have done this in Michele Migliore's group in Palermo and in collaboration with many, many people from all these institutes that you see here. And I just thank you for your attention.